Assalamualaikum and good day to everyone and honorable judges and chairman. I would like first of all to thank the organizers, Universiti Malaysia Kelantan UMK and MAS Association of this conference and for having us here. Let me start by saying a few words about my background. My name is Ain Atika from Faculty of Bioengineering and Technology UMK. Before getting into the presentation, I would like to credit Dr. Wee Seng Kyu, who demonstrated expertise in microbiology, and Nick Amelia, who's among the first batch to explore and conducting this project. Without further, I'm very happy to present our research project entitled Effect of Shewanella on MR-1 on Microbial Influence Corrosion in MIC. In my presentation, I'll be focusing on four main parts: the introduction, materials and method, results and discussion, specifically on bacteria growth curve, Corrosion analysis, and SEM. Finally, conclusion and recommendations. Corrosion can be a huge topic or issue to discuss here, so we narrow down a bit the type of corrosion we're focusing, which is influenced by bacteria, also widely known as microbiologically influenced corrosion in MIC. What is MIC? MIC can be defined as biodegradation of metallic materials influenced by metabolic activity of microbes. MIC affects to almost industries including oilfield and marine underwater systems. It is postulated that the little thing, microbes, had colonized the pipeline surface during crude oil transmission, subsequently damaging the metal surface through pitting. Pitting corrosion referred to formation of small holes on metal surface which caused by the bacteria. At this current situation, MIC is responsible for 100 billions on the annual corrosion costs worldwide and accounted between 20 to 40% within the cost. This figure shows 2016 net losses facing by oil and gas firms in USA due to loss of impairment and corrosion mitigation control in oil and gas industries. So why Shewanella on MR-1? This facultative anaerobe of Shewanella is capable of reducing a wide range of electron acceptors, such as soluble and insoluble iron-3 metals, nitrate, nitrite, manganese, and such. MIC acceleration by iron-reducing bacteria (MIRB) are mostly through removal of ferric oxide on steel surface. But iron-reducing Shewanella also postulated to inhibit MIC by means of transforming insoluble iron-3 oxides to other iron-3 or 2 substance such as magnetite, hematite, and vivinite, which that serve as physical barrier within the microbial biofilm formation against corrosion under anaerobic conditions. So this would be our main research gap to investigate Shewanella MR-1 on metal corrosion. Preparation of Shewanella seed culture consists of incubation of a single colony from agar plates and incubated overnight at 30 degrees Celsius at 150 revolutions per minute on shaker. There were two media used for corrosion assessment. The one is LB, Luria Britannia, a rich carbon media, and minimal salts M1 media. For minimal salts M1 media, a liquid of 40 millimolar HFO was amended as the sole electron acceptor. This is how we designed the ionel corrosion assessment. As you can see, here's an incubation vial that was filled into 60 milliliter of either LB media or M1 media. All corrosion assessments were conducted in triplicates to attain accuracy. Throughout the experiment, the corrosion of iron 2 as the product of iron 3 were monitored via fluorescein assay. The purple color indicates a positive result, while colorless solution indicate a negative result. The positive results were measured at 562 nanometer absorbance via spectrophotometer. And finally, the corroded iron nail were taken out and cut prior to surface topography analysis via scanning electron microscopy. The growth curve of bacteria show hyperbolic graph. When lag phase from 0 to 3 hours, the population of bacteria then increased exponentially from 6 to 24 hours. Underwent log phase. At this phase, Shewanella experienced rapid growth and high dividing state. And then after that, at 27 to 30 hours, the bacteria underwent a stationary phase where there will be no longer increase in Shewanella cells population. Lastly, the number of bacteria started to decrease continuously from 32 hours, indicates that the Shewanella underwent that phase. Ferric reduction activity were monitored via fluorescein assay to quantify the dissolved ferrous ion incubated with iron nail in M1 media with different concentration of yeast extract within 30 days of iron nail corrosion assessment. B5 refer to biotic samples supplemented with 5 gram per liter yeast extract show the highest increment in ferrous concentration compared to the abiotic sample A1 supplemented with 1 gram per liter yeast extract. Overall, based on figure 3, both abiotic and abiotic condition in LB media show the highest ferrous concentration compared to M1 media. These results suggest that LB media is a rich media culturing bacterium containing tryptan, yeast extract, and sodium chloride for Shewanella consumption growth with iron nail incubation, while M1 is a minimum media composed only minimal ingredients to support Shewanella growth. The sign label A showed an irregular dense mineral layer with numerous edges in which it showed the characteristic of iron oxide. The sign label B showed cracks. This suggested the mineral are the product of corrosion, probably iron oxide. As you can see, crack structure were observed in both abiotic and biotic conditions. A flower and folded-like structure was formed, referred to level C under both conditions. Level D suggests corrosion layer that has been corroded from pitting corrosion. The presence of crack on the surface of iron nail are due to the pitting of corrosion product. Technically, as the ferrous iron being reduced to ferric iron being reduced to ferrous, the insoluble ferric change its state into soluble ferrous. So the soluble form of ferrous aid the pitting process, causes the iron nail surface to be cracked. As a conclusion, this research suggests that Shewanella oneidensis MR-1 enhance corrosion process by the formation of corrosion product on the iron nail surface. The crack on the surface also provide evidence there is pitting corrosion had occurred. Action of iron 3 was correlated to the corrosion process with the increasing of iron 2 concentration. Therefore, the activity of Shewanella oneidensis MR-1 to microbial influence corrosion is correlated to each other. That are commonly used are identifying the surface topology of iron nail under scanning electron microscope (SEM). The image from SEM has to be also supported by X-ray diffraction in bracket XRD, by which the corrosion product can be identified and get proven through analytical assessment. Also, it also suggests to use carbon coupon steel instead of iron nail due to its flat surface, so make it easier to observe under SEM.